The project conducted was investigating global diversity in the plant genus Bacopa. Bacopa consists of about 60 flowering plants that vary in morphology. Many grow in North and South America, while a few are found in Africa and other parts of the Eastern Hemisphere. The U.S. currently houses 7 species in this genus. Bacopa is broken down into 4 sections to further categorize its characteristics. The sections are Brahmia, Ketodiscus, Herpestis, and Mela. We hypothesize that each section in Bacopa will be confirmed by our molecular data as monophyletic, and that the morphological traits of each section will support the molecular phylogeny. This part of the poster identifies some common characteristics of Bacopa sections, including height, number of flowers per axil, veins, and bracteoles. We can see here that Bacopa grows mainly in North and South America, while in parts of Eurasia and Africa too. Our project also tries to understand how Bacopa dispersed to Africa. We can note that within each section, there may be only one species that lives in Africa.
For this experiment, we extracted DNA from plants in the genus Bacopa and compared the sequences for 2 genometric regions, the nuclear internal transcribed spacer and the chloroplast NDF gene, using phylogenetic methods. We investigated the DNA data for as many species of Bacopa as possible, and evaluated the boundaries among species and categories that were established using morphological data. We extracted DNA using a standard CTAB protocol. Following the extraction, we conducted polymerase chain reaction, or PCR, to amplify the ITS region, using primers designed by Cheng, and the NDHF regions, using primers designed by Olmsted and Sweer. We then sequenced the cleaned PCR products using Sanger sequencing. After sequencing, we used our DNA sequences and previously published sequences to build a phylogenetic tree for Bacopa by aligning the DNA sequences to each other and conducting a phylogenetic analysis in IQ tree. The plant morphology of many Bacopa species were compared and contrasted, giving insight to what plants are more closely related to each other.
In this figure, we show the phylogeny of Bacopa. We constructed this using combined DNA from the nuclear ITS region and the plastid NDHF gene. We reconstructed geographic dispersals into Africa and highlighted them. As you can see, there are 4 separate ancestral dispersals into Africa. There are 2 in section Mela, 1 in section Ketodiscus, and 1 in section Herpestis. We found that Bacopa eugenesis is in fact the same species in Africa as it is in the Americas. We found support for sections Brahmia, Ketodiscus, Herpestis, and Mela. We also found that sections Ketodiscus and Herpestis are closely related, also sections Brahmia and Mela. Morphological characters largely supported the sectional classifications, although there is some uncertainty that still remains.
In the results, we discussed the hypothesis that at least one of the dispersals of Bacopa into Africa could be closely related to species in Central and South America, as well as species in Southeast Asia. This aligns with our results as seen in the taxonomic table here, as each of these highlighted regions into Africa all have closely related species in Belize, Thailand, Mexico, and Brazil. You can also see evidence of this in our dispersal maps here, 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 and here, where each of these species has, plants living in sections of Central and South America, as well as Africa, Southeast Asia, and some of them. In future research, we would continue to gain insight into the evolution of this plant species and perhaps even find new subsections of life in, Southeast Asia, maybe Australia regions, and so on and so forth.